we think we have a tiger by the tail, something very, very unique, very, very different, and very exciting, and not just exciting to us, but the general oncology community as well.
BioNeex is the first biopharma R&D open exchange platform for licensing and co-development partnerships of drug candidates. We are working closely with biopharmaceutical companies that are actively seeking preclinical and clinical stage drug candidates for in-licensing and co-development partnerships to expand their R&D pipelines. If you are a development stage biotech company and are looking for licensing and co-development partners for your drug candidates, then list them for free on BioNeex and get contacted directly by these biopharmaceutical companies. We are already working with many partners. Would you like to learn more about BioNeex? Feel free to get in touch with us. We look forward to working with you.
Hello everyone, today we are back again with BioNeex interview. Our guest is today co-founder and CEO of Oncolyze, Dr. Stephen Evans. Hello Dr. Evans, it's great to having you today.
Hi Simbat, it's delightful to be here. Thanks so much for the invitation.
Please, Dr. Evans, tell us about Oncolyze and what makes this development stage biotech so unique.
Thanks Simbat. So Oncolyze is developing a drug which has a new mechanism of action against a newly identified target. So this is a drug, and we'll get into the details and the technical aspects of it a little later, but fundamentally, this is an approach which has not been tried in patients before in this way, but the preclinical data that we have has shown that we have tremendous potential of this drug to treat many different types of cancers. And along the way, we've accumulated a really terrific management team. We have the top KOLs in our particular target field, which we'll discuss again a little bit later, not only on board, but incredibly enthusiastic about getting this drug to market through clinical trials and to market as soon as possible, because there's a real potential that this drug can actually end up helping, I don't want to overblow it, but millions of patients. So we think we have a tiger by the tail, something very, very unique, very, very different, and very exciting, and not just exciting to us, but the general oncology community as well.
Yeah, thank you, Dr. Evans. I know the Oncolyze leadership team, and they are highly experienced, coming from the industry biotech side. They brought a couple of companies already to IPO, so we definitely can rely to your leadership and of your co-founders. Could you please tell us the story so far with your drug candidate OM301, and what is it, how it works, and what are the potential indications so far for that one?
Sure. So this is a really fascinating story and deserves a couple of minutes of attention. So we actually identified the technology. It was kind of a science project, if you will, at the time we found the technology. It was a downstate medical center in New York City. And what we saw, we were so excited about the potential of this technology to be turned into a deliverable drug that we actually formed the company specifically to license the technology from the downstate medical center and go down the essentially very arduous pathway of turning a molecule into a deliverable drug. So the actual drug itself was developed, interestingly enough, in silico by computer modeling. The inventor of the technology, who was at Downstate Medical Center, was the chairman of pathology there, wanted to develop a small peptide that would go intracellular and attack the intracellular pathways and cause a cell to die by apoptosis. So, standard killing a cancer cell by apoptosis. So of course, it's not easy to get a peptide inside a cell because you've got the cell membrane. So you need something to lead the peptide, which is not soluble in lipid, across the cell membrane. So he had an idea to attach a kind of a Trojan horse, what's called an amphipathic molecule. It's a well-known type of leader sequence which can lead a peptide across the cell membrane. He developed it in silico. He modeled it in 3 dimensions. He got it to look like he wanted it to look. He had some of this peptide synthesized in the laboratory, and he gave it to cancer cells in vitro. And the cancer cells died, but they didn't die the way that he expected them to die. They didn't die by apoptosis over a several-day period. They died almost instantaneously by lysis and necrosis. Now, I like to say here that a lesser scientist would have said, this is something wrong with my hypothesis. This is a toxic molecule. I'm going to go back and do something else. He sensed something was there. Pasteur said, chance favors the prepared mind, and Pasteur won the Nobel Prize. So I think this is a Nobel Prize in medicine if this gets into clinical use and it gets to take care of patients. And what he discovered was that, and it took 10 years in his laboratory of research to finally publish the paper that documented this. What he discovered is that his drug was not going inside the cell and interfering with cellular mechanisms and pathways. It was actually, and this is a total fortuitous discovery, was actually attaching to a cell surface molecule, which is found on the surface of cancer cells, but not normal cells. And when it attaches to that cell surface molecule, that cell surface protein, the binding part, which is the part that was targeted to go intracellular, actually binds extracellularly. And then the amphipathic leader sequence, that Trojan horse, sits there and it pokes holes in the cell membrane. It porates the cell membrane. And when you poke holes in a cell membrane, the cell expands osmotically and it bursts, it dies, it explodes almost instantaneously. So he discovered a whole new target and mechanism of action completely fortuitously. And by 10 years of hard research in his lab, came to understand the mechanism. So it turns out, and this is even really more interesting in a sense, that the target that he was going after, which is intracellular, is a well-known intracellular oncoprotein. It's HDM2. And many, many companies over the years, as I'm sure you know, have tried to drug that pathway intracellularly and failed. Nobody knew that HDM2 was highly expressed on the cell surface of cancer cells, as well as being highly expressed intracellularly. So his molecule fortuitously was binding to its target, but its target was in a different place on the cell surface, and it was causing lysis and necrosis of the cells. So that's the background of how this discovery came to be and what we found so fascinating about it. And it turns out that HDM2 is expressed on the cell surface of almost every cell cancer line. We've looked at over 25 different cancer cell lines, and it's expressed on the cell surface of all those cells, all those different cancer types. But it doesn't appear to be expressed on normal cells on the cell surface. So based on that, we formed the company to license the technology and take it forward. And we've had really fantastic preclinical results since that time.
As any biotech who is in an early stage, I think Oncolyze might also looking co-development partners, to move forward this drug candidate. Can you a little bit tell us about that? Who would be an ideal partner? And indeed, are you looking for co-development partners? And I also want to tell our audience that Oncolyze drug candidate is already on BioNeex. And Steve, why you believe then BioNeex also can help you on that? Thank you.
Right. So interestingly and importantly, the drug can target both solid tumors and hematologic cancers, blood cancers as well. So the potential to target multiple different types of cancers is there. So in terms of development, we know that many different companies, large pharma, mid-sized pharma, have specific interests in either things like solid tumors or in hematologic tumors. So we think that there is a great potential for a co-development partner in either the solid tumor space or the hematologic malignancy space as well. So we think that there are a large number of potential folks that we could work with in a co-development pathway. We can certainly, and we're planning, to move ahead with our GLP toxicology and submit our IND to FDA and to commence our first clinical trial, which will be in acute myelogenous leukemia. But at any point along the way, we would really welcome a co-development partner because, of course, co-development brings a tremendous amount of imprimatur as well as good funding and potential for a wide distribution and a more rapid pathway. So, essentially that's something which would be very favorable. Look, we're pretty agnostic in terms of how we move forward. We want to bring value to our investors, and we want to bring this drug to patients as well. So we have two very, very important goals, and the best way that we can get there is the way that we will select to do that. We came to BioNeex, I think fairly early on in BioNeex's history, and it's grown tremendously rapidly since that time. But we saw tremendous potential for the type of service that BioNeex provides as a marketplace, if you will, connecting small companies and startup biotech companies with larger companies and potential partners who otherwise we would not be talking to, or we would not even have an in to get into. A lot of the larger pharma really want to see a phase 1 clinical trial before they'll even pick up your call and talk to you. So BioNeex is a kind of a unique platform in that sense to get a more widespread knowledge and interest in the drug.
Thank you, Stephen. Indeed, we are having a strong focus on BioNeex, not only like on big pharmas, but also the mid-size clinical stage biopharmas who are seeking, who are interested in a preclinical stage assets. And this is where mostly the biotechs are struggling, so they can done their IND enabling studies and then go to clinical trials. And that's where we come in, connecting with the potential clinical stage mid-sized companies specifically. So what is the next milestone, Steve, that you as a CEO now leading the company to reach?
So there are, of course, many milestones along the way. We've selected AML, as I mentioned earlier, acute myelogenous leukemia, as our first clinical target. We have a really, really deep and very exciting preclinical data set showing that the drug can not only treat circulating cancer cells in the blood, but also treat the AML stem cells as well. We treat cancer and cancer comes back most of the time. And many times it comes back because even though we can treat, if you will, the adult cancer cells, the cancer stem cells are a renewable population and keep returning as well. So our preclinical data was very, very strong. We collaborated with City of Hope Hospital in California, MD Anderson, and have found that the drug actually attacks the cancer stem cells, the AML stem cells in the bone marrow, while leaving normal stem cells alone. Now, that's a really exciting finding because if you can get a drug that's, a lot of drugs will treat cancer and treat the cancer stem cells in AML and suppress the heck out of the bone marrow. If you can find a drug, hopefully ours fits this bill, that has a very low, if any, toxicity, off-target toxicity profile, this could be a tremendous winner. So that's why we're going after AML as our first clinical indication. The milestones would basically be, of course, getting through our GLP tox, getting our IND, and starting the first clinical trial, the phase 1/2A clinical trial. We have our clinical sites picked out already, our principal investigators, our KOLs. So we've got, if you will, a lot of the car built, and now we just need to get some gas into the engine to move ahead with the IND enabling studies and then into the phase 1. So those are the two big milestones coming up. IND enabling studies, getting the IND, and then getting into the phase 1/2 trial.
Thank you, Stephen. Very good to know. And I know the team is already did such a work in the past. It's not the first time that you guys are doing, and what are the pieces of the puzzle to putting together. And that was, I would say, the short-term, mid-term goal. But what is the long-term vision for Oncolyze. Can you tell us a little bit about that?
Yes, so I'd be happy to. So, of course, you're absolutely right that the short term and the midterm is to get into clinical trials and show safety and efficacy of the drug in this particular patient population. The next step will be to expand the clinical indications one by one, and then go for, we already have a secondary indication, which we're working on now for multiple myeloma. We have lots of good preclinical data in solid tumor as well. So the mid to long range is to then move this drug from hematologic malignancies into solid tumors as well, and expand as much as possible the patient populations. Also, this was a drug that was developed in silico and has now become, a deliverable drug. I'm very enamored personally and professionally and scientifically by the in silico approach. I think there is a tremendous potential for developing new drug, new chemical entities, both against our target and potentially against other targets as well. So as we become successful, and hopefully we'll be extremely successful, we'll be able to have more of a budget for research and development for other types of chemical entities as well, and expand the whole concept of this type of targeting with small peptides against cancer targets with low toxicities to normal cells and to patients.
Yeah, it sounds for me, you develop a kind of a platform, right? In biotech world, they like to call it. That means you don't only target one indication, that's just a start, but you see there's a enormous potential for other indications too. And this, I think, what pretty much the other side of the industry, like big pharmas are looking for, that they can produce more and more like drug candidates for other indications, right?
Right, that's exactly our plan. And assuming we're successful as we go forward, that's near and dear to my heart.
That's great. Thank you, Steve, for this great interview, talking to you. I always give a chance to our CEOs for the last question. Is there anything else that I didn't got the chance to ask you? Could you please use this last 1-2 minutes for that?
Well, I would say that the one thing that really, it may not distinguish us from other biotechs, but it distinguishes us in our own minds, in any case, is that we are a team which is completely passionately driven by the ability to bring something to market that cures patients. We're physicians and we're scientists. We have been working so far without salary, and that will change upon our large funding, but this has been a dream of passion. And as a doctor, if I do a few procedures a day and I save a few lives a day, this is very great. But if I can bring a drug to market that saves millions of patients, potentially, how great is that? So this is the passion that drives our team, and we're all completely behind this in terms of our corporate culture and our own personal passions and desires.
Thank you, Steve. I'm always fascinated by the CEOs who are medical doctors, because being on one side as a scientist, bioengineer, or a biologist or pharmacologist, yeah, you understand. But then really the doctor who knows the pain that patients are having for such a devastating disease like cancer, that's really should be passion to run a company without really taking even a salary. Thank you so much for your work, Steve. I personally appreciate it. I want to remind our audience that today our guest at BioNeex interview was the CEO and co-founder of Oncolyze, Dr. Steven Evans. And Oncolyze is developing a novel therapy that selectively attacks cancer cells while leaving normal cells intact. Thank you so much. Thank you, and we will come again with another BioNeex interview.
Thanks so much, Simbat.